Hi, everybody. Welcome to Pediatric Grand Rounds. So excited to have everyone in the room and everyone who's here online with us. We have a big crowd today, so super excited. I'm going to turn things over to Dr. Nielsen, who's going to introduce our speaker and our lectureship for today. And here's Dr. Nielsen to do the rest of the hard work. Thank you.
Good morning, everybody. Thanks for coming. Just as a reminder, so we're the Global Health Collaborative. So if you haven't heard of us, we are a department committee. And so as part of our committee, we sponsor one Grand Rounds a year. So last year it was our own Dr. Laura Ellington, who talked about her work. And we're thrilled to have a visiting professor, Dr. Teresa Quartz, today. So just a brief review. So our Global Health Collaborative, the mission of this organization is really to connect people doing global health work, since there is a ton of global health work within Seattle, but a lot of it is somewhat siloed. And so we're trying to connect clinicians with researchers to try to advance global health initiatives. And here's our leadership team. So I don't want to take any time from Dr. Quartz. So it is my absolute pleasure to have Dr. Teresa Quartz here. She is a friend and colleague from the University of California, San Francisco. She is a University of Washington alum. She completed her undergraduate and medical training here at UW before leaving here and going to Stanford to do her clinical training in pediatrics and pediatric critical care medicine. Then that was not enough for her, so she went on to get her MS and her PhD in global health sciences from UCSF. And she is currently an associate professor of clinical pediatrics in critical care medicine and the director of the Institute for Global Health Sciences affiliate program, as well as the director of the Pediatric Global Health Scholars Pathway at UCSF. So in addition to that, in the past, Dr. Quartz has been a sepsis consultant for the WHO and is currently the PCSAM lead for the UCSF WHO Collaborating Center. She's also a past co-chair of POLICI, which is a pediatric critical care research organization, the global health subgroup, where she led an international study to describe the burden of pediatric critical illness in resource-limited settings. And finally, she is the current member of the SCCM Sepsis in resource-limited settings task force. So as you can see, Dr. Quartz is an expert in pediatric sepsis in resource-limited settings, and I'm thrilled to hear about her work today. Let's give her a warm welcome.
Thanks, Katie. It's really a pleasure to be here. So as Katie mentioned, I am a UW alum, so I have to say this is actually where my love for pediatrics started as a volunteer in the emergency department. And I remember being in this auditorium for grand rounds and many presentations. So for me, this is really kind of coming full circle. So it's a real honor to be here, and I appreciate all of you spending your time in your morning here. So with that, I want to tell you a little bit about the work that I've done. And so this is entitled From Bedside to Bench and Back. And so I really do a clinical translational work, and I hope that you can see that thread throughout. I have no disclosures. And so I also want to, going through what we'll talk about today, there's kind of like two broad areas of work that I focus on. And one area is this global collaborative work, which I've done with Katie and Amelie. And so I'll talk about the Global Parity Study, which is a study that we designed to measure the burden of pediatric critical illness in resource-constrained settings. And I have my own personal research program as well. And that's where we're using multi-omic approach to better understand pediatric sepsis in Tanzania. So I'll kind of switch gears in the middle, and I'll warn you when it's coming so that you know that we're going to change our focus. So first, with the Global Collaborative Project, Global Pediatric Acute Critical Illness in resource-limited settings. So as Katie mentioned, I'm a pediatric critical care physician. So that means that actually the majority of my clinical work is trying to avoid child death, right? But I work clinically in San Francisco, but that's not where the majority of child mortality is occurring. In fact, 80% of global child deaths are in resource-constrained settings. And you can see that immediately when you look at a graphic like this, which is showing the under 5 mortality, all-cause mortality, for children under 5. And it's very evident where blue is low rate of mortality and red is high, where the child mortality is occurring. To put some numbers to this, the under 5 mortality rate in the US is 115 per 100,000 population, but in South Sudan, that's 3,000. So that's just a staggering number when you think about it. So when I see data like this, my next question is, okay, then what are children dying from? And this graphic is showing the top outside of the neonatal period, under 5 causes of death globally. And it's bucketed into infectious diseases, non-communicable diseases, and trauma. And I look at this, again, from the lens of a critical care physician, and what I see is infectious disease-related deaths are essentially sepsis, right? And if we think about how do children die from pneumonia and tuberculosis, well, it's probably hypoxic respiratory failure. Diarrhea, hypovolemic shock, electrolyte abnormalities, and for trauma, it could be multi-organ dysfunction, hemorrhagic shock. These are things that critical care physicians treat regularly. So maybe there is a role in averting childhood death globally for critical care physicians to play. So following on that, I wanted to better understand, and our group in general within the Policy Global Health subgroup, what the burden of critical illness is globally in children. And there have been a couple of foundational global pediatric prevalence studies to help us better understand that. But there's a few limitations to them when we looked at it. So the first one is, if you look at the studies we've listed, they focus on a single disease entity. So it's acute lung injury, respiratory distress syndrome, sepsis, and neurologic insults. And so when you focus on just a single entity, you kind of lose the bigger picture of what the burden of overall critical illness is. And for clinicians in the room, you know that it very rarely is just cut and dry that you're treating only one thing. Like, how many times has a child that has pneumonia developed ARDS and then also has sepsis, right? So there's this overlap between these conditions. And so we're really, by focusing just in this siloed approach on a single condition, missing the overall estimate of critical illness. The second kind of gap we identified is that if you look at the geographic location for most of these studies, it's primarily North America and Europe. Very few low and middle income countries are represented, if at all. And so that begs the question whether the data from these studies is really generalizable to those more resource constrained settings, which, as I've shown, is where the majority of child mortality is occurring. And then the final thing is the sample population. So classically, we define critical illness as admission to a pediatric intensive care unit. That makes sense. But the problem is that pediatric intensive care units are not universally available. And so a lot of critical illness globally is actually managed outside of formal ICUs, on the ward, in emergency rooms. And so we're missing that entire population of children if we define critical illness by a location in a hospital. And so that really is a problem with not having a definition for critical illness that's resource independent. And so with that in mind, the Pediatric Acute Lung Injury and Sepsis Investigators Global Health subgroup set forth to try to kind of answer some of these questions. And so we had 4 major objectives. The first was to develop a definition for pediatric acute critical illness that could be applied across resource variable settings, so wasn't location dependent. The second was then to take that definition, actually measure the prevalence and etiology of pediatric critical illness. The third was to generate data for resource allocation and policy level decisions. And the final was to create a global pediatric critical care research network that was inclusive of resource variable settings. We had noticed that most of the studies, as I showed on the last slide, incorporated only European and North American sites. So how could we bring in those resource variable settings as well? So with that in mind, to tackle the first objective, we used a modified Delphi approach to develop a research definition and a framework for acute pediatric critical illness. And through this process, this was led by Anita Arias out of St. Jude, we defined acute pediatric critical illness as a risk for life-threatening deterioration, and there were 2 main domains with 2 subdomains in each. The first one was acute physiologic instability, which could be measured by either abnormal physiologic parameters or vital organ dysfunction or failure. And the second domain is a clinical support requirement, which could look like either a need for frequent or continuous monitoring or a need for time-sensitive intervention. And you see, nowhere in here is anything about admission to an intensive care unit or any other location in the hospital. So with that definition in hand, we were then able to set out and measure the actual prevalence and etiology of pediatric acute critical illness, specifically in resource-constrained settings. So we recruited globally, and ultimately we were able to recruit 46 sites that represented 19 countries and over 7,500 children. And what you see in this map, outlined in the bright colors, are the global burden of disease regions, and then the countries that participated in the number of sites per country are shown. The gray scale is showing the sociodemographic category. And so we decided to use a sociodemographic index as a measure of socioeconomic development because it's a metric that takes into consideration the country income level as well as fertility rate and educational attainment. So it's more of a holistic measure of overall development. It's also been shown to be associated with health outcomes. And so with that, this was a point prevalence study, so we had 4 different time points throughout the course of the year. And we did a 24-hour sampling frame, which meant that every child who presented with an acute illness or injury during a 24-hour period, whether that was through the emergency department or a direct admission, was screened for inclusion. And the participating sites were resource-constrained hospitals. Well, that can mean many different things. So we had every participating site fill out a very detailed hospital resource survey so that we could compare apples to apples. And this was a non-neonatal pediatric population, so outside 28 days to 14 years, in children with acute illness and injury, so not coming in for vaccinations or suture removal, etc. And we defined acute critical illness using the definition previously developed, but operationalized it in the following way. So within 48 hours of presentation, again, the acute piece, acute instability resulting in death, a clinical support requirement, which was one of the domains, and that was operationalized as need for non-invasive or invasive mechanical ventilation or vasoactives, or a need for increased monitoring or time-sensitive intervention. This we operationalized as need for admission or transfer to an ICU or HDU or another facility outside of the hospital for a higher level of care. And then we followed admitted children for up to 30 days for outcomes. And overall, we found that the prevalence of pediatric acute critical illness was 13%. I'm going to say it in a different way because I think it sounds a little bit different when you say it as 1 in 8 children who sought care who had acute illness or injury to a participating hospital had acute critical illness at the time of presentation. And so could you imagine if that was the situation here, if 1 in 8 kids coming through the emergency room door had critical illness. So that's actually quite a staggering number when you think about it like that. We also found that there was a higher prevalence in infants, as well as children who were presenting to sites in low sociodemographic category, and children who had pre-existing conditions like sickle cell cancer and congenital heart disease. And on this graph, I'm showing the prevalence of pediatric acute critical illness on the left-hand y-axis, and on the right-hand y-axis is the all-cause mortality. Across the x-axis is the country sociodemographic category, and the dashed lines are showing what the cohort proportions were across the entire population. And so when we break down the prevalence of acute critical illness and mortality by sociodemographic category, we see, again, this trend towards a lower sociodemographic category having the higher burden of disease and mortality. And in general, that decreased as the sociodemographic category increased. One of our other objectives was to identify the etiology of critical illness. And so what we have here is a ranking of the most common diagnoses amongst children who had pediatric acute critical illness. And the higher the rank, the redder the color. If it's not in the top 10, it's gray. And so you can see that across the cohort, that's the global estimate, pneumonia, sepsis, and acute malaria were in positions 1, 2, and 3. But if you separate it by sociodemographic category, that's really being driven by what's happening in the lowest sociodemographic category. And as soon as you move outside of that, it's actually injuries that become the most important. And that's really critical information to have if you're trying to decide how to allocate your available and even limited resources, or if you're making policy level decisions, where are you going to put your efforts into? The other key piece that comes out of this is if you look at that list, again, pneumonia, sepsis, malaria, these are acute critical illnesses that do not require ECMO. They really don't require high-tech interventions. It's oxygen therapy, it's fluid resuscitation, and acute stabilization. And so we really do have those pieces already to be able to manage most of the pediatric critical illness that's out there. And so that's this concept that basic critical care is actually an essential component of the healthcare system, as opposed to something that is a luxury that only a few people can afford to have. We're actually very fortunate right now because this, I think on the tails of COVID and because of an initiative out of WHO, there's really an understanding that critical care needs to be integrated into the acute care pathway and to ensure that all people have access to basic critical care services. So the WHO has a current global mandate to strengthen emergency, critical, and operative care, and they're doing that through multiple different initiatives, one of which is they have an implementation arm through this Acute Care Action Network that societies and non-governmental organizations can join to help with the implementation piece. And then there are other initiatives as well. So Society of Critical Care Medicine is developing guidance for sepsis in low-resource settings. And then the Essential Emergency and Critical Care Global Group essentially believes that all people should have access to essential emergency and critical care, and has actually defined what is included in that and has developed tools for implementation. So with that, I'll switch gears and we'll go from the macro to the micro. And I'll tell you a little bit more about the work that I do and how we're trying to use this multi-omic approach to better understand pediatric sepsis. And at first, we just need to understand what our diagnostic and prognostic challenges are. So there was a landmark paper that was published in 2020 from the Global Burden of Disease, and we knew that sepsis was a global burden before that, but we didn't have great estimates to put behind it until this was published. And so now we can say we understand that 49 million estimated cases of sepsis per year, and 2 out of 5 of those are occurring in young children. And sepsis-associated deaths account for 1 in 5 deaths worldwide, which accounts for almost 3 million young child deaths every year. But one of the key things that I actually took out of this, and many others did as well, was the fact that 85% of both sepsis cases and sepsis mortality are occurring in those low and middle income countries. So again, that map that we showed at the very beginning, it's all concentrated in regions like sub-Saharan Africa. So I have a couple of study sites in sub-Saharan Africa. One of my main sites is in Tanzania. And so to better understand the context in Tanzania, it is a fairly populated country. There's 60 million people, but 20% of the population lives in poverty. Many people are without access to clean water and sanitation services. And there's a healthcare worker shortage. So there's 1 physician per 100,000 population. To put that in perspective, in the US, we have over 300. And there's a high under 5 mortality rate. And because of the Global Burden of Disease study, we now have an estimate that 50% of all pediatric deaths are actually sepsis-related deaths. So the burden is quite high. So the initial study that we did was focused on just understanding how children with sepsis present and what their clinical outcomes are. And so this was a prospective cohort study that we conducted at Muhimbili National Hospital. And that's the tertiary care center for the entire country. It's located in the capital, Dar es Salaam. And so if you want any type of specialized pediatric care, this is the only public hospital that can reliably offer that. And we define sepsis as clinical SIRS criteria. Overall, we screened almost 10,000 children and enrolled over 2,000. And in this cohort of hospitalized children with sepsis, the mortality was 11%. Again, that's pretty substantial. But we also found a couple of other interesting things. So the first was that it was actually very hard to predict which of these children were most likely to die. And that was probably in part because there was insufficient data to calculate many of the scores that we rely on. So the PIM and the PRISM is something that we use often in the PICU literature. But that data just wasn't there. We tried to derive a clinical score-based mortality prediction model, but it also didn't perform very well. So we're just lacking the ability to really identify who those high-risk children are. And then the second major kind of lesson I took from this was that there were diagnostics that were available, but they were pretty limited. They weren't used consistently, and they were insensitive. And so primarily it was malaria testing and then microbial culture. And a major confounding factor that limited the effectiveness of microbial culture was that 42% of this cohort actually was receiving antibiotics before they even had the blood culture drawn at outside facilities. So that makes it really difficult to trust the culture results, and the likelihood of having a false negative is pretty high. So if we looked at the likely infectious source in this cohort for sepsis, ultimately only 9% had a definitive pathogen identified. And also to point out, in this cohort, which was defined by having sepsis criteria, only 35% had any microbial culture done at all. Well, it turns out we are not the only people who have found similar results. So this is a large systematic review that looked at prospective studies that included both adults and children and assessed fever etiology using lab-based case definitions. And they found a similar in-hospital case fatality rate of 12%, and also that antimicrobial use was quite common kind of broadly across the population. And even in this systematic review where lab-based case definitions, which have more advanced diagnostics than Muhimbili had available, no pathogen was identified in 80% of cases. So it makes it very difficult to provide targeted, effective care if you don't know what you're treating. And so there's clearly a diagnostic challenge here in pediatric sepsis, and it's multifactorial, right? Like some pathogens are not easy to culture. Viral infections often go unrecognized and untreated. This pretreatment issue with antibiotics, in many places antibiotics are unregulated and you can get them over the counter, makes microbial culture, which is the mainstay of diagnosing sepsis, pretty ineffective. And then some more advanced methods like serology or PCR, you really have to have an a priori suspicion as well as the capacity to do it, so that all of that is quite limiting. So I think there are many barriers to providing targeted and effective sepsis care for children, and they're not unique to sub-Saharan Africa or resource-constrained settings, but they are exacerbated in those settings. And so distilling it down, there are many more than what's listed here, but the ones that I chose to focus on are the fact that there's an incomplete knowledge of what the etiology of sepsis is. There's also insufficient identification of who the high-risk population is. In these subgroups, there are children who could benefit from being transferred to a referral hospital. Perhaps they need increased monitoring. While PICUs are not universal, there are more regional PICUs that are being formed. And there are children who may benefit from specific therapies. So you think of a child who's hypoglycemic or anemic, there's direct therapy that you can give, but it's a time-sensitive intervention. And so I was wondering, like, how could we use kind of the cutting-edge science that we have available at an institution like UCSF to address some of these diagnostic and prognostic challenges, but also importantly, we have to be able to translate those findings back into something that we can actually implement in a resource-constrained setting. And so with that, we started to form this idea of how could we use the multi-omic approach to better understand some of these and address some of these challenges. So the first kind of omic I want to try to sell you on is metagenomics. And so on the tails of that prior study, we then designed a new study, but focused more on the sepsis etiology and testing prognostic biomarkers. Similar population, children with sepsis in Tanzania, same institution, so the tertiary care facility in the capital of Tanzania. But this time we leveraged not only the local expertise, but also those across UCSF and the Chan Zuckerberg Biohub. Same population, 28 days to 14 years. We performed locally available diagnostics on all children, so it wasn't up to clinician discretion. So we had microbial culture, malaria testing, and HIV screening on all patients. And then we measured point-of-care biomarkers locally, and then we collected a fair amount of specimens so that we could do additional testing. So we did plasma so we could test biomarkers, and do additional pathogen detection, whole blood for transcriptomics, and pathogens as well. And overall, we screened over 15,000 children and enrolled 755. Very similar to the last cohort. When we applied our locally available diagnostics, again, we had malaria testing and microbial culture. And in this cohort, actually 52% of patients had received antibiotics prior to arrival. Same kind of finding. 12% of children had an identified pathogen. And so that's where metagenomic next-generation sequencing, I think, can be a real tool. So metagenomic next-generation sequencing is an unbiased pathogen detection technique, and it's unbiased because it detects nucleic acid from any type of microbe that's in a patient sample. And a patient sample can be blood, it can be a nasal swab, it can be a tracheal aspirate, it can be CSF. And so nucleic acid is extracted, whether it's RNA or DNA, and then essentially the ultimate goal is to prepare a sequencing library and to sequence those fragments of DNA or RNA or complementary DNA. At that point, the sequencing data then passes over to what we call as the dry lab. It's a bioinformatics pipeline. The one we use is CZID-seq, which is actually open source and free online. And through a series of very complicated bioinformatics steps, the bases are called. There's a lot of quality control steps that are that it has to pass through. The human reads are removed from the sequences, and then the remaining sequences are compared against reference databases for sequences of known microbes. And with that, that alignment occurs for what is like a likely microbial hit. But just because a microbe is there does not mean that it's a pathogen. So there's one more critical step, and that's where a lot of the kind of clinical adjudication comes in, is applying what a rules-based model that was developed by a collaborator of mine, Dr. Langelier, for actual pathogen identification. This is a pretty cool thing, so I'm going to go into it in depth. So after the sequencing data passes through the pipeline, the output that you get is a heat map like this. And what this is showing is that each column is a patient, and each row is an identified microbe. And so what you can see here is the microbes are actually ordered in order of abundance, and so the abundance is measured in reads per million, anywhere from 0 to 2,000. And so you can think about it as the reads per million being the strength of the signal. So the redder the box, the stronger that microbial hit is. And they're ordered in order, as I said, of abundance. But you can see that one patient actually has several potential pathogens, right? Like they might have a hit for E. coli and Klebsiella. So now how do you distinguish between them and say which one you think is the actual pathogen? And that's where the rules-based model comes in. So it identifies the disproportionately abundant pathogen, and whether that's bacteria or fungi within a sample, and identifies the greatest gap in abundance. So here, E. coli is ranked number 1. Another potential pathogen is ranked number 2, but you can see that there's a giant gap in terms of which one is more abundant. And so it identifies that gap in abundance between the top-ranked microbes, and then it compares the microbes above a particular gap to a known reference database, and then calls it as a likely pathogen or not in the sample. For DNA viruses, it's actually more simple. It's just based on the abundance. And so applying these methods and metagenomic next-generation sequencing, we actually identified a likely pathogen in 66% of children as compared to 12%. And importantly, when we compare it to the blood culture results, there were 149 potential bloodstream infections that were missed by culture. So these are the culture-negative sepsis. You can also see that there are some viral infections in there, as well as parasitic with the schistosomiasis. So one of the biggest criticisms, though, of metagenomic next-generation sequencing is that it's resource-intensive and it's not available in real time. And I agree, that is very true, and we use it even very rarely in high-income settings. But I would argue that I think that we're not that far away from being able to use this technology in real time, and it's because of a couple of things. One, the cost of sequencing is decreasing pretty steadily over the years, and two, because of initiatives like this. This was a partnership with the BioHub and the Bill and Melinda Gates Foundation, which put low-cost sequencers in low and middle-income countries so that they could do the sequencing locally, upload the data to the cloud, and then run it through the pipeline. So I don't think we're that far away from being able to use this technology in the foreseeable future. We also applied the same idea, same concept, but for whole genome sequencing to identify the antimicrobial resistance genes. And so this is, instead of taking a patient sample, we took a blood culture that was positive, identified one of those isolates, so now we're just dealing with one bacterial isolate strain, extracted the nucleic acid from that. Library prep sequencing, similar to the prior path way I shared, goes through also the CZID-seq online, but it's a different reference database for antimicrobial resistance. It's the CARD database, and then the output from that is identifying antimicrobial resistance genes. And we were able to construct this antibiogram. And so what this is showing is each column is an antibiotic and each row is a patient, and it's clustered by the microbe or the bacteria that was identified. All of these are gram-negatives. The gram-positives are not shown. And if it's susceptible to the antibiotic, it's green. If it's not susceptible, it's red. And so it's very obvious without looking even too closely that there's a high rate of resistance. And it's even more concerning than that because when we look at the antimicrobial sensitivity testing, both phenotypically and by the antimicrobial resistant gene identification, they're mostly ESBL. And when we looked at the antimicrobial resistant gene, you can see CTXM, which confers ESBL, is in almost every single one of them. And that's one of the most common antimicrobial resistance genes around the world. What we also discovered, and it's not shown here, is that we actually had two patients who were admitted 2 years apart from each other who had almost the identical microbe sequenced, which means that there is ESBL E. coli that's circulating in the community. These are not hospital-acquired infections. The other thing that was quite concerning was the resistance to ceftriaxone, which is currently the first-line antibiotic for children with sepsis and severe febrile illness, both at the hospital level and across in the national Tanzanian guidelines. So this is one of our kind of first big win, I think, was that we were able to present this data locally, and the hospital revised their sepsis protocol. And so what was before ceftriaxone for everyone regardless now has become, if a child has previously been treated or is not improving, you should move to a second line. We can talk about whether meropenem is the correct second line or not offline, but at least we now have the recognition that maybe ceftriaxone shouldn't be the first line for all children. So the next kind of omic I want to address is protein biomarkers, and so sort of the proteomic angle. So we tested biomarkers, and we wanted point-of-care testing, again, because we wanted something that was potentially deployable at the bedside. And so we selected biomarkers for which we had data and for which there was already a clinically approved test. And so we chose C-reactive protein, ferritin, procalcitonin, and lactate, and all of these have been shown to have increased levels associated with either mortality or illness severity. We also selected hemoglobin and glucose because, as I mentioned previously, we want to be able to identify the children who would benefit from time-sensitive interventions like blood transfusion or dextrose. So we implemented this iChroma 2 machine, which is able to turn around the CRP, ferritin, and procalcitonin within 20 to 30 minutes for patients. And so that was done locally in Tanzania. And then we had an iStat machine for the lactate and hemoglobin, and then just a regular glucometer. And when we looked at the differences in the biomarker concentrations between the survivors and non-survivors, they were all statistically significant. And so in general, worse or higher in the non-survivors. But the one that really stood out because of the order of magnitude difference between them was procalcitonin. Now, I'm not going to show you the data for all of them, but we measured the area under the receiver operator characteristic for all of these biomarkers for mortality. And the one that performed the best was procalcitonin. So procalcitonin as a single biomarker and nothing else had an area under the curve of 0.8. And then we added some relatively easy to measure clinical signs, so difficulty in breathing and altered mental status, and we were able to improve the area under the curve to 0.88. So you can see how procalcitonin, with a couple of physical exam findings, could really strengthen the ability to triage these high-risk children. Certainly performed better than our other models had. As I mentioned too, we did collect plasma. This we shipped back to UCSF and did additional biomarker testing. And here we were interested in kind of understanding the immunopathology of sepsis a little bit more. And so we looked at the pro and anti-inflammatory biomarkers that have previously been indicated or implicated in sepsis, as well as endothelial injury, fibrinolysis, and leukocyte adhesion. And we calculated the area under the curve for these single biomarkers. Again, we're trying to find this idea that maybe there's a biomarker out there that we haven't identified that could actually be even more sensitive and specific. And in general, they didn't perform great, but soluble TREM1, which is the soluble triggering receptor expressed on myeloid cells, performed the best. Again, kind of matching what procalcitonin did at 0.8. Well, not like that doesn't blow you away, but I will say I was excited to see this for two main reasons. So one is because this has actually been shown to be a significant biomarker in other quite diverse populations. So this is a population of Ugandan children where soluble TREM predicted in-hospital mortality with an area under the curve of 0.89. And in a completely different pediatric population in Southeast Asia, soluble TREM again predicted death or vital organ support within 2 days with an area under the curve of 0.86. So now we have this biomarker that's showing that it's significant in different febrile pediatric populations. The other reason why I think it's quite exciting is the fact that it is a biomarker. It plays a role in inflammation amplification and innate immune response. But there's also a potential therapeutic target. So nangibutide is a synthetic inhibitory peptide fragment. It binds to the TREM1 agonist ligand. And there was actually a recent phase 2b randomized control trial in adult patients with septic shock. It was a relatively small sample size. And while it didn't show a statistically significant improvement in SOFA score, which was their primary outcome, there was some evidence that the high-dose nangibutide could actually result in greater improvement in the patients who had a higher soluble TREM. So I don't think this isn't ready yet, I think, for prime time, but I think that things like this are actually quite exciting because it allows us to, one, identify the high-risk patients, and two, there's actually something potentially actionable that we can do about it. And then finally, for the last omic, the transcriptomic. So there is a role, I think, also for host gene expression signatures. So a fair amount of work has been done in US-based pediatric sepsis cohorts. For those of you who are familiar with Hector Wong's work, much of it has come out of work that he has done. And host gene signatures have been shown to be associated with sepsis severity, a complicated clinical course, multiple organ dysfunction syndrome, and mortality. So our question was, is that also applicable in an African pediatric population? It hasn't been tested before, but it has been looked at in an adult Ugandan septic population. So this is hospitalized adults in Uganda with sepsis. And they used whole blood RNA-seq, so bulk RNA-seq, and then assigned participants based on their gene expression signatures to sepsis endotypes signatures that had been developed in high-income countries. And what they found was that there were some shared biological and clinical features between the high-income country cohort and the Ugandan cohort, but there was actually very poor agreement overall between the classifiers. And that meant that over half of the participants actually switched which endotype they were in. And the authors of the Ugandan study hypothesized that that was probably because there was a high prevalence of co-infections in the Ugandan cohort. Not surprisingly, 50% had HIV, tuberculosis was common, as was malaria. And these are pathogens and co-infections that we know can alter the host immune response to infection, as well as the fact that their treatment, which often includes steroids and long-term antivirals, can also affect the immune response. The other thing that was a key difference was that there are major differences in the host pathogen profiles. So to say this in another way, what's causing sepsis in Uganda is not what's causing sepsis in Europe. It seems obvious, but it's worth saying out loud. And so with that in mind, we then did a differential gene expression analysis in our cohort. And what this is showing is a volcano plot where we compared survivors to non-survivors, and the genes that are upregulated in the children that died are shown in orange, whereas those that are downregulated are shown in green. The genes that didn't meet the significance cutoff of 0.05 are shown in gray. And while the differential gene expression analysis identified 800 genes that were differentially expressed between survivors and non-survivors, there were two clear outliers that you can see in that upper right-hand corner. And so the first one is WACh, which is an enzyme that mediates fatty acid production. It's also been shown previously where higher levels of WACh are associated with higher mortality, and specifically with more severe respiratory infections like COVID and RSV and influenza. And then the second outlier was IL-1R2, and that's a receptor protein that inhibits IL-1 signaling, so it dampens inflammatory effects. So we were curious, if we took these two gene outliers, how well did they, individually and in combination, actually predict mortality? And it turns out WACh as a single gene achieves a similar area under the curve, 0.81, and the figure is shown here, as procalcitonin by itself and soluble TRIM. And if we combine WACh and IL-1R2, we actually get an area under the curve of 0.84. For comparison, if we put all 800 differentially expressed genes into one model and also include some key clinical risk factors like age and malnutrition, the best area under the curve we could achieve in this cohort was 0.85. So a two-gene classifier is actually quite predictive and powerful. And again, while not quite ready for prime time, there is a potential for a simple prognostic classifier like this, which is host-based, that it could be translated into a clinically practical qPCR assay. And there is, in COVID, a bit of a legacy for doing that. And so you could see how that could potentially be feasible in a resource-constrained setting. So where do we go from here, and how do we pull it all together? So kind of recentering on this idea of, how can we use this amazing science and technology that we have at our disposal in high-income countries and at the universities that we're at to address some of these larger challenges? And then also to always keep in mind, how to keep it relevant and feasible. So I think that we're, with this work, taking a step towards providing targeted and effective sepsis care for children. And the next major step is external validation. And so that includes other pediatric populations within Tanzania and throughout sub-Saharan Africa, but also looking at geographically diverse regions as well as adult populations as well. So is this a signal that's just specific to this population, or is it more generalizable? And so that work's in process right now. And then the big picture, and really the ultimate goal, is to be able to develop a treatment algorithm in which we can incorporate the local sepsis pathogen epidemiology and the antimicrobial resistance patterns, and then incorporate the point-of-care tests to be able to assist with risk stratification. And this screenshot is from the EPOCT tool, which is a tool that was designed and implemented for outpatient pediatric Tanzanian patients to identify the children who needed antibiotic therapy and needed referral to a hospital. And you can see a model like this that incorporates the clinical data, clinical diagnostic testing, as well as risk stratification, and then the output is one of a diagnostic and treatment recommendation. So that would be the end goal, is to be able to make those context relevant treatment algorithms. And then I just want to end with kind of a thought for all of us to think about, and that is that where a child is born can determine whether they have access to clean water, food, and education, and really can determine their overall ability to access good health. And I would like to argue that I think we're in a very privileged position with where we are, and that we should use the resources that we have at our disposal to try to change that arrangement. And so I don't think that geography has to determine destiny. And that's where I think some of the work that we do can really try to move the needle and impact it on a greater level. And so with that, I have many people to thank. Global Parity was a huge project, and there's other co-PIs, Adnan Butta and Adrian Holloway, who are very involved in every step of the process, as well as the steering committee, and then the investigators themselves, and then the team in Tanzania, without whom none of this would be possible. And then multiple collaborators throughout the Langelier lab, including Van Pham, who did the differential gene expression analysis, and Padmini, who did the metagenomic next generation sequencing, and then multiple other collaborators and mentors. And so with that, I'd like to thank you for your attention and your time. I'm happy to take questions, and please fill out this evaluation.
That was indeed a fantastic presentation. Thank you so much. I have two questions for you. I was wondering, do you think that the sequencing data could inform a priority designed qPCR testing that could be used that might be less resource intensive? That's one question. And then the second is regarding the biomarkers that predict mortality. So if a child presents to a hospital and with critical illness, and child A has a biomarker that's highly predictive of mortality, but child B, who has the same clinical presentation, does not, how would you determine differences in treatment? Would you? Or, so that's my question.
Such great
Thank
questions. Yeah. So the metagenomic next generation sequencing we're really using for pathogen identification. So we're using that in a couple of ways. So the main one is just to understand the epidemiology of sepsis better. So more in like a surveillance method, and so that we can inform empiric guidelines or antibiotic selection. I think in terms of like a diagnostic test, there we're actually looking more at the host response. So is there a host-based response that can help us diagnostically distinguish between viral, bacterial, and parasitic? And so their previous work has actually shown that there are signatures, host signatures, that can distinguish bacterial from viral. It's never been looked at in a population that can also have parasitic infections. So that's actually the direction we're going in terms of the diagnostic tool, as opposed to using the metagenomic sequencing. And then with the biomarker, it's such a good question, because I didn't get into this, but in sepsis, there's a lot of evidence emerging in both adult and pediatric literature that there's these subphenotypes of sepsis. And so there are kids who look very similar, but have different outcomes and different responses to therapy. And so ultimately, if we can identify what those subphenotypes are, and then identify them with a combination of point-of-care biomarkers and clinical signs, then maybe we could actually, for those two kids that look identical, be able to see the one that would benefit from a particular treatment, like steroids. Or maybe steroids is actually worse for some kids, and we should avoid it at all costs. So I think that's the direction we're going to go in next, is to better understand those subgroups of sepsis patients. It's exciting, well, to me.
Thanks, Teresa. That was really amazing. So thinking about the resources you need for a high-quality microbiology lab, and then the resources you need for doing this kind of point-of-care omics, how do you imagine that evolving and being the most effective resource utilization in the next several years, like realistically, then also ideally?
Yeah, well, maybe this is just a pipe dream, but you know how in many places they just skipped over having land phone lines and just went straight to cell phone? Like, maybe that's what we should do in some cases. And so if we could develop a host-based test that's more sensitive, specific, and timely than microbial culture, which, as we've seen, has a lot of false negatives, then maybe we don't spend the effort and resources into developing that, and we instead focus on more of the point-of-care tools. So that would be, again, kind of the 10, 20-year vision.
I love the leapfrog technologies. I was going to also kind of a follow-up question comment to what Donna was saying about identifying the kids who have those subtypes. It seems like there's also potential, if you have a country with 2 ICUs and 8 beds in each, and these kids could be identified, and potentially even transported, or even within the same hospital system be put in the right place because they're at bigger risk of deterioration. Do you see a role for also sort of focusing the resources on those kids who have the biggest risk of mortality.
Exactly, and a good example of this is actually Muhimbili, because they do have a PICU, so there is actually an actionable step you can do when you identify these high-risk kids. And so you can say, you can fast-track them, or, you know, save that last PICU bed for this one. So it is harder when you're in the more rural, peripheral regions, but in places where transport is possible and a PICU or higher level care, then identifying those kids as quickly as possible to start that process is key to success.
Thank you for the incredible work. I'm curious to zoom out a little bit. As a general pediatrician, I think a lot about the first thing, data point that you showed us around resource-limited children in poverty and coming in and having the highest rates of mortality across the board. I'm curious about the kind of gene analysis and if the possibility that this is an epigenetic phenomenon where you have the resource limitations and the things that are making that child predisposed to be sicker interacting or upregulating a gene expression in some way that is impacting their ultimate outcomes, and if there are things that can be done to reduce those risks in the broader, I mean, many of which are much, much lower kind of resource intensity,
Absolutely.
But are certainly part of what allows people to survive.
Yeah, it's a really good point. And, you know, all of these studies are hospital-based. So I guess 2 points about that. Absolutely, like there are modifiable risk factors much prior to that. I don't study epigenetics, so I don't feel like I can comment on that. But a good example of what we have seen is that there's actually a higher rate of gram-negative rod bacteremia in the population in sub-Saharan Africa than what we would see in at least what we see in San Francisco. And that also kind of correlates with the higher rates of malnutrition and this cycle of diarrhea, malnutrition. And so I think that there's probably more gut translocation that's happening. So if you, speaking about like downstream, that's where it's really important what the sanitation and water sources are, and then also the vaccination. So you're absolutely right. Like moving it even further, right? By the time they get to the hospital and they're already bacteremic, we've missed multiple different opportunities to intervene from a public health standpoint. Oh, the other thing I was going to mention too is that one of the studies that we're doing right now in Wanza, we've also, one of my collaborators has done extensive work on the post-discharge mortality. So these are children that are going home, have, you know, seemingly recovered from sepsis, but their mortality rate is quite high in the first 6 months after discharge. And so why is that? And so he's focused a lot, this is Dr. Weens out of UBC, on trying to understand that relationship. And so we've partnered with him so that we can actually collect some of these samples at the time of discharge and then correlate it with their long-term risk of out-of-hospital mortality to see if there is some signal there. We're also doing repeated sampling in the community so that we can understand some of these like bigger trends.
Hi, thanks again for this amazing talk. A question that I have is a little bit more methodsy maybe, but I guess from like an implementation perspective or even just like a research and validation perspective, like why do you choose to use something like this next-gen sequencing over something like qPCR for the pathogen identification that is possibly cheaper and like more labs maybe have the capacity to do so? Like why pick the next-gen sequencing?
Yeah, in part because it's completely unbiased. And so with one sample, like we can pick up everything that's there. And that's actually one of the biggest challenges of metagenomic next generation sequencing is it's so sensitive that you get a lot of noise. And that's where that rules-based model is really key to be able to identify the pathogen. And so we had, my mentors had done this in another cohort in Uganda and had actually found some pretty surprising things, including novel viruses that hadn't been described previously. So there's also the potential that there are pathogens out there that haven't been previously described that you would miss if you didn't have an unbiased pathogen detection technique.
Hi, thank you. Reiterating how wonderful this is. You're speaking my love language. Post-discharge mortality, AMR, risk stratification, so thank you. My question is about your AMR results, and if I understood correctly, you were using your like genes found on whole genome sequencing and translating that to an antibiogram that may or may not be used in Tanzania. And I'm involved in a lot of research where we have AMR genes detected by qPCR and sometimes whole genome sequencing, but it's always hard for me to think about what's the translation of that to these low resource settings where often there's not even routine culture susceptibility data, and there's such limited antibiotics. Like, we wouldn't want to state, like, just because there's a resistance gene to ceftriaxone that should completely
come off the
thing, because that's better than amoxicillin or something.
Right.
So yeah, just curious, like, how you're thinking about that, how it's received in country, and how you're towing that line.
Yeah, so I kind of glossed over it, but the phenotypic antimicrobial sensitivity testing was done locally, just using disk diffusion method. And so what we did to create that antibiogram was actually the combination of the two. So the phenotypic locally conducted antimicrobial resistance testing plus the antimicrobial resistance genes. So because of that, it was done locally, and so I think it was actually well received because it was done by one of the microbiologists, and he was actually a huge champion for getting the protocol changed as well.
I have a related question to Patty. So how do you balance when you find in vitro resistance with making recommendations for antibiotic change when maybe in vivo that antibiotic is still highly effective?
Oh, very good question. And I am a critical care physician, so I am going to claim ignorance. But I will say that for the antibiogram, we did not find discordant results, at least in this cohort, but did prioritize the phenotypic antimicrobial sensitivity testing over the genomic results.
We have 2 questions from our online friends. Joshua Bress, fantastic talk. We're
Thanks, Josh.
seeing a similar trend in other areas of global health, moving away from broad syndromic approaches towards more tailored therapy that reflects local epidemiology and the individual patient. It's a great step forward. It also makes the work more complex and harder to teach. From what you've seen, what helps clinicians successfully make that shift in settings where syndromic management has been the norm?
No, that's a really good question because I know, right? Because here we're saying that a child who looks exactly the same might actually have a very different clinical outcome and may benefit from different treatments. So I think it's actually a shift away from the assumption that it is a syndrome and understanding that sepsis is really more of an interaction between both the host and the pathogen, and so we have to understand both of those pieces. It's so different. I mean, thinking about what sepsis is in sub-Saharan Africa versus what we consider sepsis and at UCSF, those are completely different entities, and we're dealing with different comorbid conditions, different co-infections. So I think it's actually moving away from the syndromic approach where everyone gets IV fluids, IV antibiotics, blood culture as quickly as possible, and actually doing more of that tailored therapy. And I know we don't talk about it very much in resource-constrained settings, kind of that personalized medicine, but I don't think that it's that far off again to be able to use some of these point-of-care tools to be able to tailor therapy appropriately to the population and the child in front of you.
Great. And then finally, from Kafo, one of our colleagues in Kenya, thanks for the excellent job you and your team are doing in Africa. The sepsis subtypes you're talking about, is there a possibility to identify them by their clinical presentation?
Oh, what a great question. So I didn't include this, but we have identified 3 subphenotypes in this cohort, which have essentially like different inflammatory profiles based on the biomarkers. And those include both the plasma biomarkers that are not available point of care and those that are. And so there's kind of a high inflammatory, a medium inflammatory, and a low inflammatory subgroup, and they have different outcomes and clinical presentations. And so the very next thing that we want to do, it's just not ready to show you, is see if we can identify those phenotypes with just clinical signs and point of care biomarkers alone. I don't know if it's going to be possible, but that's the end goal. And so if we could do that, you could see how there would be the ability to identify those kids clinically and then act on it. So invite me back in like 10 years or so.
On my calendar.
Thank you.
Thanks, everyone, for coming. This was amazing. And if you were excited about this, we have Dr. Courts for many other events today. So I was just going to pull up a slide. You can just find me or find us down here and follow us because we're going to be going to FA2 where we're going to have a little networking event with our colleagues from WACh. And then after that, we're going to have several faculty presentations where we'll hear more from Dr. Quartz and also from some other faculty who are doing excellent work here. And then we will have some of our fellows, both Sarah Mayer, who is here, and Dr. Samuel Kafo, who just asked that great question. So come down here if you want to join us for that. And finally, I just wanted to thank Sophie Whitsett, who I think stepped out, but she helped us coordinate all of the logistics for Dr. Quartz's visit. So as we're going, can we give a big round of applause to Dr. Quartz?